Understanding DNA replication in prokaryotes. Steps of replication in prokaryotes. 1. Initiation. 2. Helicase loading and unwinding. 3. Single-strand binding proteins. 4. DNA G primase and priming. 5. DNA polymerase III and leading strand synthesis. 6. Lagging strand synthesis and Okazaki fragments. 7. Termination. 8. DNA gyrase and supercoiling. 1. Initiation. DNA replication in prokaryotes begins with the binding of DNA to the origin of replication, ORIC. ORIC consists of a 245 BP long at-rich sequence which is highly conserved in almost all prokaryotes. Mostly two types of sequences are present in this region, 3 repeats of 13 BP called as a 13-mer and 5 repeats of 9 BP called as a 9-mer. A higher concentration of protein A signals to start cell division. Protein A, DNA A protein, with ADP, binds to the 9-mer sequences of ORIC. This binding allows the opening of 13-mer sequences of ORIC by bending the DNA. This binding leads to the unwinding of the DNA strands, forming a single-stranded region called the DNA unwinding element, DU. This unwinding is facilitated by DNA C protein. The DNA C protein assists in loading DNA B helicase onto the single-stranded DNA. Helicase loading and unwinding. DNA B helicase, also known as the G protein, is loaded onto the single-stranded DNA by DNA C and other factors. It uses ADP hydrolysis to further unwind the DNA at the replication bubble. As the helicase unwinds the DNA, it exposes the single-stranded DNA templates for new DNA synthesis. Single-strand binding proteins, SSBs. Single-strand binding proteins, SSBs, attach to the exposed single-stranded DNA regions, preventing the separated strands from reforming double-stranded structures. SSBs also protect the single-stranded DNA from degradation by nucleases. DNA G primase and priming. DNA G primase, an RNA polymerase, synthesizes short RNA primers complementary to the single-stranded DNA templates. These RNA primers provide a starting point for DNA synthesis by DNA polymerase III. DNA polymerase III and leading strand synthesis. DNA polymerase III is the primary enzyme responsible for DNA synthesis during replication. On the leading strand, DNA polymerase III adds nucleotides to the growing DNA strand in the 5' to 3 direction, using the RNA primer as a starting point. Since the leading strand is synthesized continuously in the same direction as the replication fork movement, DNA polymerase III can work continuously. Lagging strand synthesis and Okazaki fragments. On the lagging strand, DNA polymerase III synthesizes short fragments called Okazaki fragments, each starting with an RNA primer. After the synthesis of an Okazaki fragment, the RNA primer is removed by the exonuclease activity of DNA polymerase I. DNA polymerase I replaces the RNA primer with DNA nucleotides and fills in the gap. DNA ligase then joins adjacent Okazaki fragments, creating a continuous lagging strand. Termination. Replication continues bidirectionally until the replication forks meet at a specific termination site, terc. Termination involves specific termination proteins that assist in disengaging the replication machinery from the DNA template. DNA gyrase, G protein, and supercoiling. DNA gyrase, also known as topoisomerase II, is a G protein that alleviates the supercoiling of DNA ahead of the replication fork. As the DNA is unwound during replication, it creates torsional stress ahead of the fork, which can lead to supercoiling. DNA gyrase introduces transient breaks in the DNA strands and allows them to rotate, relieving the torsional stress.